So let's draw out what this actually looks like in molecular terms. And we're going to add in some proteins that are playing a role. And so we have a piece of DNA. And there's going to be a site on that DNA that we're going to refer to again as the origin. And in some organisms, these are very clearly defined by specific DNA sequences. In us, that's really not so much the case, and yet there's going to have to be features that help the cell recognize these in some way. And then you have to have something that binds to that. You have to have some DNA binding protein that can come in and recognize that site. And so there's a protein complex called ORC, for origin recognition complex, whose key job is to recognize that site. So this group of proteins actually was found by Steve Bell, who is a faculty at MIT, and he actually also teaches a molecular biology course that you can see online and take if you're an MIT student, which is a pretty fascinating place to play. So we have this origin recognition complex called ORC, and it's going to associate with other proteins as well. So for example, a protein called CDC6. So this is going to bind to the DNA, and so here we have a state where we're in G1. We can see these events happening, and we're going to now start building things onto this. Okay, so we have here ORC, same with the origin, and it's going to bring in a complex that's going to help us actually trigger things. Okay, and this is a group of proteins that's going to actually circle DNA. So this is going to be the machine that ultimately does the job of unwinding and unfolding the DNA so that the polymerase can get in there and start attacking things. It's a hexamer of proteins, and so we'll refer to this as the MCM hexamer or the MCM helicase. Okay? And this is going to come in with a helper protein called CDT1. And so by bringing this in during G1, not only are we marking this site, but now they're bringing in this hexamer to encircle the DNA. And so it's actually not enough to just do this once. You have to bring another batch of proteins in. And really beautiful work from Steve Bell's lab and others have tried to understand the mechanisms by which the cell is being able to achieve this and coordinate this. But ultimately, you end up with a situation now, again, where you have the ORC still there, but there are two of these rings now, both encircling the DNA. And so when this whole process is triggered, they can go in opposite directions. So in this case, this helicase is loaded. It's all ready to go, but it's not active. So it's been loaded during G1, and it's sitting there waiting to go, but it's not really ready yet to be able to achieve this. Okay? So we have this in G1. And now we're going to come through to that synthesis phase where we're actually going to make the DNA. And there's going to be things that basically now say, hey, go. It's time to be able to start. I'm going to fire you. And means turn you on, not eliminate you. And so there's going to be two regulatory enzymes that help for this, two kinases. One called CDK2 cyclin E. And we're going to come back to these players so we can discuss a little bit more about how they work. And another kinase called DDK. And so these two kinases are going to act at the origin to trigger this. But this also needs to serve to restrict further activity. So this is going to allow us to fire the origin, but it's also going to phosphorylate the loading factors, the things that help it get there. And so these two proteins, CDC6 and CDT1, are also going to be phosphorylated. And they're going to do something that actually is very similar to what we discussed in the last batch of signal transduction lectures, which is that they're going to use this phosphorylation as a signal. And so in this case, phosphorylation allows the cell to further recognize these using an E3 ligase called the SCF. So this is an E3 ubiquitin ligase. And as you remember from the signal transduction lectures, this is going to add a protein modification called ubiquitin to these proteins, and it's going to target them for degradation. So this is a pretty dramatic thing to do, but this provides an irreversibility that basically you cannot reuse them. And because you cannot reuse them, they're not going to be able to act to add more MCMs on. And so we can use the MCMs that we loaded during G1, but then we're not going to be able to trigger this again in any way. Okay. And in this case now, we're going to get a situation where we're going to have this sort of bubble. The DNA is going to be unwound, and so what you're seeing here is the two strands apart, and these MCM helicases are going to be on either end of this, continuing to unwind the DNA. And now the polymerase can act here to be able to copy in the DNA and continue. So every time we copy the DNA, we're going to go in both directions. We can unfold it. We're going to travel one way. We're going to travel another way. And as the polymerase does that, it really actually needs this hexamer to continue to unfold the DNA as it does that. And then finally, during G2 and mitosis, we really don't want to do this again. We've degraded them, but obviously they could be resynthesized in some way. And so there's a protein called giminin. And this is an inhibitor of CDT1. And so this is going to be expressed, and so that it just can't come back and be active again. So this is a CDT1 inhibitor. Okay. And obviously, if we kept that around forever, that would be a problem. And so basically, at mitotic exit, giminin itself needs to be degraded. And it's going to use another machine that we'll talk about both a little bit today and also in the next lecture, called the anaphase promoting complex, or cyclosome, that is another E3 ubiquitin ligase that will achieve this. And so it's a pretty nice cycle. We have a way to be able to load the origin. We have a way to be able to fire the origin during this S phase or synthesis phase. And then we're just not going to do it again. Okay? And coming back, we will end up monitoring this in different ways. We'll discuss the process of fluorescence-activated cell sorting, or FACS, to be able to measure that. You can actually also monitor the incorporation of new nucleotides. So there's an analog of thymidine, BRDU. And you can actually detect the incorporation of this into your DNA. It's a great way to measure DNA replication.